Your immune system stopped one of your own cells from developing into cancer and saved your life by quietly killing it somewhere in your body. That is what it always does. By far most of malignant growth cells you foster will be killed without you of all time taking note. Which is a very difficult task because cancer cells are parts of yourself that begin to act like people, even if it hurts. What is malignant growth and how does your body kill it constantly? Introduction. Cancer occurs when damaged cells multiply unchecked. It can come from almost any kind of cell in your body, so there are hundreds of different kinds of cancer. Some develop gradually, others are forceful, some can be dealt with successfully, others are destructive. As it were, a cell that becomes diseased transforms into something old and a new thing. Over billions of years, cells have evolved to compete for space and resources in a hostile environment to survive and thrive. Until an exciting new lifestyle emerged. Participation. A division of work that permitted cells to practice and climb to a higher level together. However, collaboration requires penances. Disease cells quit being important for the aggregate and become people once more. Some errant cells are manageable by your body, but some cancer cells divide repeatedly, creating a new organism within you. Stealing the resources you need to survive, buying for your space, and destroying the organs they were a part of. In spite of the mischief they cause, disease cells are not underhanded. They will not harm you. They have no demands at all. Cells are protein robots that simply follow their programming, which sadly has been tainted. The cell's soul in a nutshell. Your cells have a DNA-filled nucleus. It comprises of qualities, guidelines for how to assemble proteins and when to make every one. In order to make proteins, these building instructions are copied and transferred to ribosomes. Your cell's capabilities are determined by the kinds of proteins they produce. Every day, tens of thousands of times, your DNA mutates, or slightly deteriorates. The majority of the time, just by being alive, there is no special reason. Practically, these changes are fixed rapidly or are not risky. However, as your cells replicate themselves over time, damage builds up. Envision making duplicates from duplicates from duplicates for quite a long time. Perhaps a hair or a corner got caught in the scanner one day. The new copies and all subsequent copies incorporate each new error. If you smoke, drink alcohol, are overweight, breathe in asbestos, don't use sunscreen, or get a virus like HPV, you can damage your DNA more. Be that as it may, the least complex method for harming DNA and persuade disease is to be alive long enough. There is no other explanation for the majority of cancer cases than bad luck. The harm that prompts disease. We are rearranging, however, generally. There are three classes of qualities that should be defiled so disease can emerge. The main key change is in the fittingly named cancer silencer qualities, or TSGs. There are many things these genes are. As far as one might be concerned, they produce control instruments that constantly examine your DNA for mix-ups and duplicating blunders and fix them right away. Additionally, they prevent normal cells from recklessly multiplying. Your cells basically forget how to repair themselves and can reproduce unchecked if TSGs become damaged. Your oncogenes may undergo the second significant mutation. At the point when oncogenes are turned on, the cell is told to quickly increase. When you were still in your mother's womb, they were extremely active. A single original cell must divide and grow rapidly in order to transform into trillions in a matter of months. When there are enough of you, these genes for rapid growth are turned off. At the point when your oncogenes get undermined, once more, they essentially turn on. The third significant transformation is in your cell's self-destruction switch. The majority of cells are recycled and renewed frequently. Apoptosis, a controlled suicide, is usually initiated by specific genes when cells accumulate excessive damage. On the off chance that the qualities that control this cycle get harmed, cells are allowed to live on notwithstanding being hazardously adulterated. As a result, a cell becomes a young cancer cell if it is unable to correct errors in its genetic code, loses the ability to self-destruct when it notices damage, and begins to grow rapidly without restraint. These cells must be eradicated as soon as possible. Despite their badness at this point, they are still relatively weak and simple to kill. But if they keep evolving and multiplying, they might learn how to get around your defenses and become a real threat. Your immune system is searching for these cells at all times. But how can corrupted cells that appear to be identical to healthy ones be identified and destroyed? Instructions to track down malignant growth. Indeed, here we return to the proteins your cells produce and the story they tell. So if, for instance, your oncogenes switch back on, they make oncogene proteins. Your invulnerable framework realizes that they ought not be available assuming you are a grown-up. Your immune system must therefore be aware of the proteins that are being produced within each cell in order to distinguish between healthy and corrupt cells. To address this advancement thought of MHC class I particles, a kind of show window that makes cells straightforward. To demonstrate what they are doing, cells frequently incorporate tiny portions of the proteins they produce into thousands of these MHC molecules. The selection is constantly updated, providing a current picture. Your immune system stores all of a library of proteins that are extremely hazardous and should not be produced by healthy cells. It has billions of T cells, which are made to recognize particular proteins. A T cell immediately kills a damaged cell when it detects a prohibited protein in an MHC display window. However, this system has a flaw. What would happen if a cancer cell mutates and discovers a way to evade this process? It will disappear completely if it stops producing MHC class I molecules. Without show windows, the invulnerable framework is visually impaired and can't recognize malignant growth any longer. Fortunately, evolution came up with a clever solution. The cell that kills by nature a jury, judge, and executioner in one. The killer at this very moment, your body is being patrolled by hundreds of millions of natural killer cells in search of cancer-causing or virus-infected cells. In order to check for one thing, natural killer cells move from cell to cell. Additionally, a cell containing a secret must be destroyed. What makes the normal executioner cell considerably more metal, is that it is dependably in murder mode. In an effort to eradicate every cell in your body, it patrols your body. Your solid cells need to persuade it that they MHC class I molecules provide a means of doing so. So in outline, practically all youthful disease cells you will at any point foster in your life will be killed by your safe framework. Okay, but why do we still get cancer if your body is so prepared? Well, cancer cells sometimes undergo more mutagenesis and become much more adept at fighting back. Disease is an account of a weapons contest. A war in the arms that we will eventually win, possibly with the assistance of natural killer cells. A number of therapies, including cancer-fighting vaccines, engineered T-cells, and even natural killer cells, are beginning to show incredible promise. These therapies will be discussed in subsequent videos. Therefore, the war is not yet over, but we are moving on to cancer, which will eventually be eradicated once and for all, perhaps sooner than we anticipate.